Hello everyone, I'm Hao Xu. I'm the tree fruit physiologist at Summerland Research and Development Center, Agriculture and Agri-Food Canada. Today, on behalf of my colleagues Daniel Ediger and Yoshi Watanabe, I am presenting this update, part 1 of heat impacts on apple that we investigated after the heat events that hit the interior British Columbia in summer 2021. Like many tree fruit production zones in the Pacific Northwest, the Okanagan Valley experienced a couple of sustained heat waves in late June and July, leading to massive forest fire and considerable heat damage on the fruits. Right after the heat waves, sunburn necrosis and browning started to show up on many apples, particularly on the southwest side of the canopy. In order to understand the mechanisms underlying the heat damages and to facilitate early evaluation of the compromised fruit quality, we conducted a study on ambrosia and gala apples to characterize the external and internal traits of the sunburn browning fruits in August and at harvest. And we also evaluated the differences rendered by rootstocks. Sunburn browning is thought to be caused by excessive UVB radiation and high fruit surface temperature above 45 degrees Celsius. Under the confocal microscope, Yoshi's lab captured the autofluorescence coming from the chloroplast and vacuoles of different types of fruit skin. In this tangential view on the left, in the cells on the outer layer of the normal skin without any damage, there is abundant autofluorescence coming out from the chloroplast, filled with chlorophyll and autofluorescence as green, and also from the vacuole filled with anthocyanin and autofluorescence as reddish purple. When the skin is moderately damaged, you can see there are still living cells present, giving out the anthocyanin signal or fluorescence signals. However, there is significant chlorophyll degradation. And when the skin is severely damaged, no living cells are found to be present. Instead, they are filled with unidentified autofluorescence stress-responsive material. In the cross-section view on the right, it is easy to see the cuticle in all three samples. In undamaged skin, there are healthy epidermal cells in the outer layer. These cell structures are less obvious or apparently damaged in the damaged two samples. And also there is an intense green autofluorescence coming from the cuticle in both damaged samples, which correspond to the signals in this tangential view. This could be accumulation of some stress response compounds like melted wax or phenolics as the fruit responds to prevent water loss from the wounded skin and to localize damage and prevent further damage to the surrounding tissues. And these changes could alter the spectral characteristics of the fruit surfaces. Using this handheld DA meter, we detected some differentiations in the fruit skin absorbance between 670 and 720 nanometer when the fruits were still on the tree prior to harvest. 100 fruits of each type were measured non-destructively. The damaged sun-exposed side of the fruit showed significantly higher delta absorbance than the normal sun-exposed side, and this is consistent in both cultivars. This change could be attributed to the altered skin texture, or the presence of the stress response compounds due to sunburn browning. This phenomenon was more pronounced in the red gala than in the bicolor ambrosia. As we know, browning can be masked off in red cultivars during coloration and maturation, so this DA meter method can be a helpful tool to detect subtle damages in the red cultivars such as gala. At the infrared wavelength, we use thermal imager to detect the maximum and minimum surface temperatures on the sun-exposed side in both sunburn browning and normal apple, and we calculated the surface temperature difference. So compared with the normal fruit, we noticed that the heat-damaged fruit had higher, larger surface temperature differences in both cultivars. This indicates the change in the heat diffusion capacity of the outer layer of the damaged fruits due to the changed physical and chemical compositions of the damaged tissues. Let's move on to internal attributes. At time of harvest, we observed lower weight of damaged fruits, which is the orange box plot, compared to the normal fruits, which is in green. And during the first to third week of ripening at room temperature, the damaged fruits also lost weight at a faster speed compared to the normal fruit. By the time of the end of the third week, the normal fruits had about 96% of weight retention, whereas the damaged fruit had about 2 to 3% less. Firmness is another important quality trait of apple. We use the fruit texture analyzer to measure the firmness on both sun-exposed and shaded sides in both normal and sunburn browning fruit. This graph showed the differential firmness between the two sides. In the normal fruits, shown in the green bars, both sides had similar firmness, therefore the difference is close to zero. However, we found an inconsistent flesh firmness inside the damaged fruit, with higher firmness on the damaged sun-exposed side than on the shaded side. This could be caused by the death of the cells under the damaged patch, or certain compound deposition increased the rigidity of the sunlit damaged tissue. When averaging the two sides, we found the damaged fruit had higher overall firmness at harvest. However, it is followed by a quicker loss of firmness during the ripening. Here is the dry matter content estimation conducted using Felix 750 produce quality meter. The damaged sun-exposed side had dry matter content higher than 20% in both cultivars, compared to the normal sunlit side, which dry matter content was about 15 to 16%. Look at the graph on the right. The fruit flesh water potential on the damaged sun-exposed side was close to minus 2.5 megapascal, which is significantly more negative than the normal sunlit side. So the both parameters, the fruit dry matter content and tissue water potential, are suggesting that the damaged tissues were experiencing some sort of water deficit at pre-harvest stage. Since the heat stress is often aggravated under water deficit, we started to wonder whether rootstocks that differ in vigor and water transporting capacity would make any differences in reducing heat damage on fruits. The results from 2021 is encouraging. In two rootstock trials, we recorded the percent of heat-damaged fruits per tree, and we noticed that in ambrosia trial, the G935, which is a more vigorous rootstocks, led to the lowest damage ratio, On the contrary, the B9, which was the smallest in the 5, led to the highest damage ratio. So similar trend was also found in the third leaf Buckeye Gala rootstock trial. More vigorous rootstocks such as G4814, G969, and G935 had lower percent of heat damaged fruits. So interpreting from the plant-water relation perspective, the larger rootstocks had some advantages in their hydraulic properties. So here's a quick comparison between G935 and B9. The G935 has larger trunk cross-section area, higher root mass, and also larger root vessel element. These trees give them higher water transport capacity to meet the water demand of the scion. When the irrigation is sufficient, better water supply of the larger rootstocks could help to alleviate the heat stress. So this suggests that the rootstock selection can be considered as a long-term measure to mitigate heat. Now you probably wonder what can be done as immediate or short-term preventative measures. You will find it out in the Summerland Research Update Part 2 presented by Dr. Kirsten Hannan and Jesse McDonald. I'd like to thank our co-op student Madeline McIntosh, USHI's technician Naoki Yuritani, and the funding support from New Tree Fruit Variety Development Council on our cap and brochure project, and our rootstock collaborators, Dr. Stefano Mozzacci, Dr. Todd Yenholm, Dr. Tom Forge, Paige Moran, Dan O'Gorman, and Melanie Walker. And thank BC Tree Fruits Grower Association and Okanagan Horticultural Advisors Group for organizing this symposium.